Globally, the prevalence of Parkinson's disease has doubled in the past 25 years. And while research into this extremely diverse neurodegenerative disorder is very active, there's much left to be uncovered about the underlying cause of the disease. Recently, Northwestern Medicine investigators have discovered novel genetic factors contributing to the risk of Parkinson's disease. These findings may serve as novel therapeutic targets in the future. Here to discuss these findings is Stephen Lubbe, Assistant Professor of Neurology at Feinberg. Welcome to the show.
Thank you. Thank you very much for having me. It's a pleasure to be on here to chat about all of my work.
Your lab investigates the underlying cause of neurodegenerative diseases such as Parkinson's disease and other movement disorders, and you do this by utilizing large-scale genomics. Tell me about this work that's going on in your lab.
Large-scale genomics pretty much involves interrogating all of the genetics, all of the DNA that everybody has inside them. So we do things like next-generation sequencing or whole genome sequencing, and then we look to see whether or not we can find changes in comparison to a reference sequence, which may be seen more prevalent in individuals with disease compared to individuals who don't have disease.
So Parkinson's disease is a major focus of your lab. How did that come to be? Tell me about that.
Parkinson's disease is a severe neurodegenerative disease where pigmented brain cells start to degenerate. And through this degeneration, you start to lose control of your movements. You start to lose control over some non-movement features and stuff. It's a severe disease that really, really costs millions of dollars to treat. I did my PhD in colorectal cancer genetics at the Institute of Cancer Research in London. And because there's Parkinson's disease in my family, I was like, hey, this is a good opportunity for me to try and make some kind of impact in my mom's life, for example. My mom was probably about 42 about my age actually, when she was diagnosed with Parkinson's disease. I found this postdoc position in Cardiff actually. And then, after Cardiff, I was happy to move back to London. And that's where I started my career at the UCL Institute of Neurology in London.
How interesting. There was a personal component there.
I feel like I'm one of the lucky few that I get to have some massive driver that steers me and guides me to try my best and to do as best work as I can. I know that my research may not have any immediate impact in my mom's life, but I can go to my mom and say, today I managed to identify some things that might lay the foundation for future therapeutic work that might help individuals with Parkinson's disease in the future. And knowing that I'm making some kind of contribution definitely helps me wake up in the morning and come to my job every day.
I want to talk a little bit about the causes of Parkinson's disease. One is past traumatic injury to the brain. There are other genetic factors as well. Describe to me what we know about the genetic origins of Parkinson's disease and what we don't know.
So to be honest, we don't know a lot about the genetics of Parkinson's disease. Very, very few people who have Parkinson's disease have an actual genetic cause for Parkinson's disease. About 5 to 10% of people with Parkinson's disease have a family history of Parkinson's disease or are diagnosed with Parkinson's disease at a very young age. Of these 5 to 10% of individuals with what we think is a genetic Parkinson's disease syndrome, we only know about 20 to 25 genes that contribute to Parkinson's disease. And these in itself only make up a very small proportion of the individuals that we know or we suspect have a genetic Parkinson's disease. So overall, we know less in terms of causes of Parkinson's disease. We know that mutations in genes like alpha-synuclein, LRRK2, Parkin, PINK1, and a handful of other genes. We know that when you get mutations in those genes, you're pretty much guaranteed to get Parkinson's disease, or your risk for Parkinson's disease is substantially increased compared to the general population. We know a lot about the common genetic variants that contribute to Parkinson's disease risk. And these types of variants have come through studies like genome-wide association studies, where we genotype hundreds and thousands to millions of common variants within Parkinson's disease patients and individuals who don't have Parkinson's disease. And then we look to see which of these variants are more common in individuals with Parkinson's disease compared to those without. And then based on this, we can estimate the amount of risk that they increase for Parkinson's disease. And generally, these common variants individually contribute about between 10 to 15% increased risk. Individually, the way that they increase risk is very small, or the amount of increased risk is very small. But when inherited together, they've been shown to potentially substantially increase your risk. For example, if you had to have the risk allele at all 90 to 94 variants, real risk for Parkinson's disease is 3 times higher than individuals who have none of those variants. Together, these 90 to 94 genetic variants that we've identified contribute about 25 to 35% of what we predict to be the total genetic heritability.
Environmental factors, as we mentioned, also play a role in the development of Parkinson's disease, and these environmental factors can interact with genetics in complex ways. Explain that to me.
Well, this is a very interesting but also very difficult topic to talk about because yes, your genetics is always interacting with your environment. You know, what you eat, what you drink, where you work, what you breathe in. These are the factors that can introduce or increase your Parkinson's disease risk. For example, people who work on farms, who are exposed to certain pesticides, their risk for Parkinson's disease is increased because of exposure to these pesticides. And similarly, people who drink water from boreholes, which contain lots of heavy metals in the water, they're also at an increased risk for Parkinson's disease. But just because you're exposed to those environmental factors doesn't necessarily mean that you're guaranteed or going to get Parkinson's disease. It's a very difficult question to answer because you can't guarantee to tell us the exact truth in terms of what environmental exposures you had, when you had them, and how long you had them for. If we can understand how genetic factors interact with environmental factors, this may help us have some kind of breakthrough in terms of offering better therapeutic targets.
And that is a goal of yours, to try to find better therapeutic targets. And according to a Northwestern Medicine study recently published in the journal Brain, you and your team have indeed uncovered novel genetic factors potentially contributing to the risk of Parkinson's disease. Tell me about this study that you detailed in the journal Brain.
I can talk about this study all day. It's one of my favorite. What we know about Parkinson's disease genetics has generally been limited to what we call single nucleotide variants. So these are variants that affect one nucleotide. But what we don't know is the contribution of more complex regions, these short tandem repeats. So a short tandem repeat is a unit of DNA that's made up of specific nucleotides, for example, CAG. And then this unit of CAG is repeated next to each other a certain number of times. When the number of repeats gets to a certain number, it can significantly increase your risk for diseases. A good example of this is Huntington's disease. The number of CAG repeat units in the general population is at a specific length. When this number repeats to hundreds of thousands of times, that's when you get Huntington's disease. So we hypothesized, hey, no one's ever looked at the contribution of short tandem repeats at a global genome level in Parkinson's disease. So we were able to utilize publicly available reference data sets and have access to genome-wide association single nucleotide data for us to kind of fill in the blanks. So what we did is rely on something called linkage disequilibrium. And linkage disequilibrium is pretty much a term that describes how certain blocks of your chromosomes are inherited together. So if we know what nucleotides are at position A, we can kind of guarantee what we know based on linkage disequilibrium, what the nucleotides are at different positions throughout that block. So we basically used single nucleotide variants to kind of impute short tandem repeats across the genome. And then we counted the number of repeat sizes in people with Parkinson's disease and people without Parkinson's disease. And surprisingly, we found 4 novel genetic variants or short tandem repeats that significantly increase your risk. And I say significantly at a statistical level. The effect size, or how much they increase your risk, is still in line with what we've seen in normal or your typical GWAS studies. They only increase your risk for Parkinson's disease by 10 to 15%. We don't know how these variants, the short tandem repeats, exactly lead to Parkinson's disease pathology. And we did some additional studies using computational-based methods to try and see if we can figure it out. So we looked to see which genes near these short tandem repeats Which genes have their expression changed by the presence or absence of these short tandem repeats? And we were able to identify potential candidate genes, which of course need to be validated and replicated in cells to make sure that we're finding the right signal. And we believe that once this is done, by whoever wants to take that challenge, once this is done, we would be able to see whether or not these new novel variants are potential targets for future therapeutic development.
That's so fascinating. You mentioned the enormous amount of data used in this study. Can you tell me a little bit more about this data?
The original data set we had constituted 16 independent GWAS studies, which we had access through the International Parkinson's Disease Genomics Consortium. This is an initiative spearheaded by Andy Singleton at the NIH. And using these studies, we managed to get data on about 22,000 individuals with Parkinson's disease and about 20,000 individuals without Parkinson's disease. And then we managed to impute STRs from a reference panel that's based on the 1,000 Genomes Project. The 1,000 Genomes Project is, as the name suggests, about 1,000 individuals where many people have looked at the genetics, they know what's going on at the genetics, so it's a pretty good reference panel. So these individuals use complicated bioinformatics algorithms to identify STRs in these individuals, and then to determine which STRs are an LD with which single nucleotide variants. So using this reference panel, we were able to impute into these 40,000 individuals to try and get a better idea of the contribution of STRs in these individuals.
You also utilize samples from the Movement Disorders Center Biorepository here at Northwestern in your research. Tell me about this resource and how does it help you with your studies?
So the Parkinson's Disease and Movement Disorder Center Biorepository is an awesome initiative that we have here locally at Northwestern Medicine. It's a biorepository where we hope to recruit individuals with Parkinson's disease and other movement disorders as they come through the door at our clinic at Northwestern Memorial. We ask these individuals, when they agree to join our studies, to provide us with blood or saliva samples in order for us to extract DNA to do genetics on them. We also ask them to provide a whole bunch of other biospecimens, for example, a skin biopsy, so we can use that as a resource to generate patient-derived neurons if we get to the point where we have a potentially interesting target to look at them. So we can generate neurons from these individuals and have a look down at these complicated microscopes that they have to see if we can figure out what's going on. The way that I use this biorepository, at the moment it predominantly is restricted to looking at families that have mutation-negative movement disorders. So these individuals come to our genetics clinic here at Northwestern Memorial because we suspect that they have a genetic cause of their Parkinson's disease or movement disorder. We highlight families that have more than one affected individual with a movement disorder, or they have a very early age of onset of the movement disorder. We then test them through our clinic to see if they have any mutations or in genes that we know are linked to the disorder that they have. And if we don't find anything, then we generate next generation sequencing on them to see if we can identify potentially interesting variants. We've managed to successfully do this on a handful of families that have come through our clinic, where we've got DNA from individuals with and without the disorder that they have within their family. And we pretty much look to see what variants we can see that are only in the individuals with the disease and not in any of the family members without the disease. And this is a very neat way to try and help us identify or narrow down potentially candidate variants. And based on the number of variants or the variants that we have, we make educated guesses about whether or not the variant that we've prioritized is the variant that we think is the cause of their disease. And using this approach, we've been pretty successful in identifying about 5 to 7 novel genes linked to dystonia and other hyperkinetic movement disorders. These genes and these mutations are exceptionally rare. We're looking at single families, single individuals that come to our clinic. So because we want to be certain that we have the right variant, we reach out to our very wide and vast network of collaborators around the world to see if they have any variants in the same gene in individuals with the clinical phenotype that overlaps with what we have. And if we still don't find anything, then we have a look at this online resource, which is called Gene Matcher. It's kind of like a Tinder for genetics. You know, you put in, say, this is the disease I'm interested in, this is the phenotype I'm interested in, this is the variant, and this this is the gene I have. And individuals who are interested in it because they've highlighted the similar thing, it kind of pops up on their screen and they go, oh, I'm interested in it, or no, I'm not interested in it. And you can swipe left or right kind of thing, and you can say yes or no. And through using this, we've been able to identify or help strengthen the evidence that we have for these disease genes or these mutations that we believe are causing the disease. And we have a handful of genes in the pipeline using this approach. So watch this space. We're going to be revealing new genes coming pretty soon.
That's really interesting. You've been able to actually solve a mystery for some of these families. What's that like to be able to sort of point to, aha, this is the genetic cause of your condition?
To be honest, this is one of the main reasons why I love my job so much. The cases that we've studied so far tend to involve children who have really, really severe early onset pediatric movement disorders. As a parent myself, I want to make sure that I do everything right and I protect my kids, but these parents are distraught and at their wits' end because they don't understand where these diseases have come from. So to be able to sit these parents down and say to them, it's nothing that you've done. You've had no control over the situation. Your child has inherited a change in their DNA that's resulted in this terrible disease. And just to see the relief in their faces and to help them come to terms with the fact that it's not anything that they've done personally, it's really rewarding for me, to be honest. And this is why I really feel that this biorepository that we have here is an amazing resource, because we see so many families like this. And it's definitely a mission of mine and the teams that we work with to try and understand the genetic causes of every single patient and every single family that comes through our door.
You've been investigating the genetic background between Parkinson's disease and malignant melanoma. Tell me about that connection.
Individuals who have Parkinson's disease are at a 2 times increased risk of getting malignant melanoma. And the reverse is true. Individuals who have malignant melanoma are at an increased risk for getting Parkinson's disease. So at a genetic level, this suggests that there's some kind of underlying genetic basis that might predispose individuals to both. And there's a whole bunch of functional data and cellular data out there that also further supports this link between Parkinson's disease and malignant melanoma. And you wouldn't think that these two, what appears to be random diseases, have some kind of link. But the link between them is that they both involve pigmented cells. Parkinson's disease is the loss of pigmented neurons in the brain, in the substantia nigra. Malignant melanoma is a cancer syndrome that involves turning your melanocytes, or the cells that control how dark or how fair your skin is, into tumors. So this is super fascinating. Individuals who have fairer skin are at an increased chance of getting malignant melanoma, and anecdotal evidence has suggested the same. The fairer your skin is, the higher your chances of getting Parkinson's disease. The pathways that produce the pigment or neuromelanin in the brain overlaps significantly with the pathway that generates melanin, or your skin pigmentation, in your skin. The genes that are involved in both, we don't know a lot about what genes are involved in both, but from the genes we do know, there's a lot of functional evidence that suggests that they may be very similar between what goes on in your brain and what goes on in your skin.
So what are you doing with that research? Are you publishing right now about this idea of Parkinson's disease and skin pigmentation and malignant melanoma?
So we are looking at individuals who have both Parkinson's disease and malignant melanoma. And we're looking at these individuals to see whether or not we can identify patterns of variation that are seen only in those individuals. And then we're taking those forward to other population-based cohorts to see if we can try and replicate those genetic signals that we're finding. Parkinson's disease is a very rare disorder. Malignant melanoma is rare, but not as rare as Parkinson's disease. So finding individuals who have both diseases is super rare, so we managed to get samples from individuals from around the world of about 100 individuals. This is how rare it is. And we're hoping that we're able to find some super interesting genetic signals. And we're hoping that what we find can contribute to what we know about both diseases, what we know about Parkinson's disease and what we know about malignant melanoma, and how we can use that information together to learn about the other diseases is super fascinating for me.
I mentioned at the beginning of the show that Parkinson's disease is increasing around the world. There is an aging population, more people are being diagnosed. Tell me what you hope to see happen in the next 10 or 20 years to help these patients.
From a genetic standpoint, I really truly believe that initiatives like the Global Parkinson's Genetics Program, the large PD project, IPDGC Africa and IPDGC Asia, these are going to play more and more important roles as we go. These initiatives are looking to recruit individuals from typically understudied populations around the world. And I really believe that they will provide a platform to try and give everybody equal access to genetic studies to understand the causes of Parkinson's disease throughout the world in all individuals of all creeds from around the world. Because we see in many different examples, what's good for one population is not good for another population. This is what I hope over the next 10 to 20 years is that we're able to make larger inroads into identifying, understanding the causes of movement disorders across the world in all individuals. Damage that's done in Parkinson's disease in the brain starts many, many, many years before you actually see the symptoms that we associate with Parkinson's disease. So over the next 10 to 20 years, I would love to be able to say we can identify individuals who are most likely going to get Parkinson's disease. So we can start looking at them early on and start administering or finding drugs that might stop the progression or the onset of the disease in these individuals.
Thank you so much for coming on the show and sharing your expertise.
Thank you for having me. It's been really fun.
You can follow us on Twitter at @NUFeinbergMed. Subscribe and ring the bell to hear about the latest groundbreaking research and discoveries. Thanks for listening.